Hi guys. Okay, so this semester we've been learning about macromolecules and cells and all of the things that go into cells functioning. Then we went through energy processing, and then we went through reproduction and genetics. And so we've been building on these ideas of what we've learned all semester. DNA biology is really kind of what we're going to call protein synthesis. One of the things that we know from learning about genetics is that your genotype codes for your phenotype. As such, your genetic material holds information, and then it is physically expressed through the cells making proteins, and through those proteins having particular jobs. When we look at DNA biology, then what we're doing is we're explaining how we go from genotype to phenotype. So this is going to be a chapter that focuses a lot on why we get these things that we get, and then sometimes we're going to talk about what happens when things go wrong. Mutations can occur, and we talked a little bit about mutations in chapter 8 with cancer, and then with non-disjunction and things like that. But when mutations occur, it can result in different physical changes. Mutations are changes in the DNA that are then expressed through the proteins as the genotype to phenotype. So we're going to dig into those particular topics in chapter 11, which is going to allow us to better understand natural selection and evolution. So let's get into it. Okay, so today we're going to go through the DNA molecule itself. We're going to talk a little bit about how DNA replicates. That is going to allow us to then understand gene expression through transcription and translation. At the end, we're going to talk a little bit about mutations. As always, you should be able to use these objectives as questions. If you need help, let me know. But let's get into it. So, we need to give credit to this wonderful lady here. So this is Rosalind Franklin, okay? Rosalind Franklin was working in a lab studying the structure of DNA in the 1950s. Female scientist in the 1950s, a very different sort of situation than today. We knew quite a bit about DNA at this point in time. We knew that there was factors involved in genetics. So we knew even prior to Mendel's time that there was some sort of inherited traits that could occur. Mendel did his work to explain that in genetics. And then we actually had the chromosomal theory of inheritance was proposed in the early 1900s, I believe 1902. So by the 1950s, we knew quite a bit about DNA, but we didn't know enough about the DNA structure to fully be able to explain how it works, how it's replicated, and how it's used. Rosalind Franklin's study, her information that she found, helped really break through and allow for others to understand this. Unfortunately, her work was basically stolen from her, okay? There was an individual in her lab, I believe it was her supervisor, who used her information, gave it to these two individuals, Watson and Crick, which we'll talk about, who then were able to use that with everything else to determine that DNA was a helix and it was a double helix. Unfortunately, Rosalind Franklin was not given credit before her death. She died of ovarian cancer at the age of, I believe, 36, and she was not given credit until much, much, much later. Without her refractal studies here, without this research that she did, we would not have gotten to this point, because Watson and Crick went together in the 1950s and 1951 to get all the data from all these different scientists, all of these different people, and try to build a model to explain the structure of DNA. Unfortunately, they did not give credit. And in science, we know that if you use someone's work, you give credit where credit is due. Without doing that, it is considered plagiarism, and it is really not okay. These two individuals did not give credit for quite a while to people like Rosalind Franklin, but their model still holds true today. And it did break through and help us understand. And they used more scientists' information than just hers. But again, you need to always give credit where it's due. Cite your sources, all right? What we've found with their stuff, their structure was just demonstrated in, I believe, 1956. And so what we know today about that structure of DNA is based on their model. This is like an infamous picture of their model. They did win the Nobel Prize for this work. Again, give credit where it's due. Okay, so what do we know about the structure based on the model Watson and Crick built? Well, we know that the structure is a double helix, and it has complementary base pairing of adenine and thymine, and guanine and cytosine. This image should look relatively familiar because this is very similar to what we saw in chapter 3 when we talked about nucleic acids. Hydrogen bonds form between the bases that are pairing up, and if you remember, hydrogen bonds are weak, but they reform easily. And so this is really fantastic for conserving information in a structure through that DNA structure being double bonded, but also having it in such a way that it can be unzipped and read and utilized. So when we talk about the DNA structure, okay, the thing that was really breakthrough, like this was fairly understood, but the thing that was really breakthrough from the Watson and Crick model was that it allowed scientists to understand how DNA replicated itself. We know from mitosis and meiosis that DNA needs to make copies of itself in order to be put into the new cells. Again, if I am making a copy and I want my new cell to have that same information as my parent cell, like I do in mitosis, I need each cell to have the same genetic information. And there was a very big question mark for a long time as to what or how this process worked. There are a lot of different theories or hypotheses proposed. The one that actually holds true and I think logically makes the most sense is the semi-conservative replication. So what happens in DNA replication during that S phase of interphase, before the cell is going through any sort of cellular reproduction, you have a single strand of DNA. That single strand of DNA is called a parent strand. That parent strand will unwind and separate by an enzyme called helicase. Helicase is going to unzip the DNA, and then there is another enzyme called DNA polymerase, which will go through and add in the matching nucleotides to the parent strand, to that kind of model strand. And so we end up with two new pieces of DNA that have half of the old information, half of the parent strand, and then half of the daughter strand, the new nucleotides that have been built. And this is how we ensure that cells have the same genetic information to go through the process of mitosis or meiosis, because this happens in the S phase of interphase. Okay, this is great for conserving DNA. It's great for ensuring that genetic material is carried over, and DNA is going to try to be as exact as possible. Now, it's not perfect, okay? If you've ever gone through and done typing and didn't have to back up and backspace and correct or autocorrect or proofread or do anything within your typing, I applaud you because you are better than I am. When you're typing, you make mistakes, okay? DNA makes mistakes when it's making copies of this strand of molecular material. It's very easy to swap out a T for an A or a C for a G or something else. There are a lot of enzymes that go through and work on correcting any of those errors that may have occurred, but it's not always 100%. And that's how we can get some of the genetic variation, even when we're talking through like asexual reproduction, okay? DNA is just holding genetic information. It's just holding the blueprints. It's just holding the materials. What really does the work is RNA. Okay, so if you remember, we talked about RNA and DNA in chapter 3. Okay, RNA does not have thymine. It has uracil. So anytime that an A would pair with a T, it actually replaces the T with a U. Okay, that's the big difference between RNA and DNA. They also have a different sugar backbone, and RNA is single-stranded. RNA does the work. It is carrying information from the DNA to other parts of the cell, and it is the process of it's the molecules actually responsible for building proteins. We have 3 different kinds of RNA: messenger RNA or mRNA, transfer RNA or tRNA, and then ribosomal RNA or rRNA or ribosomes. Remember we talked about how ribosomes were not membrane-bound organelles? That's because they are made of RNA, and so they are actually a nucleic acid. They are not, they're a little weird one. We call them an organelle, but technically are they? Not really, okay? So I do have on here some comparisons of DNA and RNA, what we should know of similarities and what we might find as the differences. And that's what we got right now. So let's look at our understanding. Check your understanding here. Make sure that if I say complementary DNA stranding, you're telling me what pairs up with these. So if you see AAC, the strand of DNA that would pair with this would be TTG, because A's pair with T's and C's pair with G's, and then we would see C, C, G, A, T, A. Okay, and that's going to be complementary DNA strand. The RNA strand would end up being different. We're going to talk a little bit more about that in the next section here. Take a minute to do these questions, and when you're ready, come on back. Okay, so the central dogma of biology is that gene expression exists. Okay, when we talk about a dogma, we talk about a central belief system, a central thing that's found in everything. All organisms go through gene expression. All living organisms today have DNA that is going to go through protein synthesis and be expressed as a protein. So the central dogma of biology is that genes are expressed through protein synthesis. Okay, this is key because what this really tells us is that DNA is made into RNA, which is made into protein. So information flows from DNA to RNA to protein. Your DNA doesn't do the work. Your DNA just has the information. Okay, there are two steps to protein synthesis: transcription and translation. Transcription comes first, and if you struggle to remember that, C comes before L, so alphabetically. Thank the Amoeba Sisters for that one, okay? DNA is going to be used to make a mRNA molecule. Okay, that is what happens in transcription. It occurs in the nucleus because that's where DNA is contained within our eukaryotic cells. And then the mRNA is going to leave the nucleus and go to the ribosome. And with the help of the ribosome and the tRNA are going to make proteins. Okay, so if you remember too early, we talked about how ribosomes are often found on that rough ER, which is continuous to the nuclear envelope. That's because those proteins need to be made effectively and quickly, and so not having to move far is very beneficial. When we talk about gene expression, another thing we have to address is the genetic code. Just like when we said that all cells have a cell membrane, cytoplasm, and genetic material, all cells share a genetic code. This is fascinating because this tells us too that very early on this was evolved and it's been maintained and conserved across all living organisms. Okay, it doesn't matter if you're a bacteria cell or a blue whale cell, you are going to use the same amino acids, and the same 3 letters of the RNA is going to code for that amino acid. Okay, there are only 20 amino acids, but there are 64 triplet codons that exist. This does build in redundancy. Okay, redundancy is important because it reduces the chances of mutations. I hope you'll notice here that if you have CU as your first 2 letters, so you have C and then U, and then it doesn't matter what your third letter is, if it's UC, A, or G, for that triplet codon, 3 nucleotides on that mRNA, all of them, CUU, CUC, CUA, and CUG, code for an amino acid called leucine. So it doesn't matter. If I have a mutation that changes out the last letter of that triplet codon, it won't affect which amino acid I'm using, so it will not affect the protein in the long run. That's important. Redundancy reduces mutations, okay? We like to call that the wobble effect because you could wobble that third letter and it still works, okay? We do have a couple of special codons that are very, very unique and are therefore not redundant. First, we have a codon called AUG. AUG is your start codon, and it codes for the amino acid methionine. All protein sequences begin with the amino acid methionine because methionine is the capital letter of the sentence. It says start here. We also have 3 stop codons, UAA, UAG, and UGA. Those are like the periods in a sentence. They tell the system that, hey, we're done. This protein is made. We don't need to make any more. The rest of these are going to be variable. They're going to be used at different points in time, and you can imagine that any arrangement of these in any length or shape is going to build out those protein sentences similar to what we have with our alphabet. Okay, so now that we know the genetic code and we understand gene expression, we're going to talk about the steps of transcription. Transcription occurs in the nucleus of eukaryotic cells, in the nucleoid region of prokaryotic cells, and it has 3 steps. The steps are initiation, elongation, and termination, because something starts, something gets longer, and something ends. Okay? Now, how do I know if I'm talking about transcription? Well, what happens in this step is going to be key. In initiation, the DNA needs to be unzipped where the gene is that needs to be copied. There's an enzyme called RNA polymerase. RNA polymerase is going to build RNA. So it's going to bind to the primer region or the start region of a gene, and it's going to begin unzipping the DNA. As the RNA polymerase moves down the strand of the DNA, we're going to add nucleotides one at a time from one of the strands, the template strand of our DNA. Once we reach the end of the gene, there is a termination sequence that will stop RNA from polymerase from going any further, and the mRNA gets processed. mRNA is processed in eukaryotes because we have a ton of junk DNA. But generally speaking, we're just like, okay, we've got the gene we need. We don't have to go any further. So we start, we add to, and we end. And in this case, it's all going to be including DNA and the nucleotides. So if we're talking about building nucleotides into a nucleic acid, we are in transcription. So what do I mean when I say that the RNA is processed? Well, we have a ton of junk DNA in our cells. Okay, this is extra credit. This is extra credit. So if you know this on the test, extra credit. Okay, put a little note, wave, star, whatever. When eukaryotes build their RNA, okay, it is very, very large because we have additional DNA in our cells. That DNA could be there for any number of reasons, but what we know is that it's not coding directly for the protein. So in order for the protein to be made from that mRNA, we have to take out the introns and leave the exons. Exons are what are expressed. Okay, once we have only the exons spliced together, we will then move into translation. And again, this is extra credit. Put a star. If you know introns and exons, it's extra credit. Okay, in translation, guess what? We have 3 steps. Can you guess what the steps are? If you said initiation, elongation, and termination, yay, gold star, okay? Because what ends up happening here is that we have initiation. We are going to start a process. Now, we are no longer starting a process involving nucleic acids. We are starting a process involving building proteins. So mRNA, well, technically we are using nucleic acids, but they're the builders. They're doing the building. They are not being built, okay? So mRNA is going to find the ribosome, okay? Now, cells are fairly large when we compare it to these smaller macromolecules. And if a cell wants to make something effectively, there needs to be a meeting point for any of those products that have to go together to make something. Pre-cell phone, I know it's hard for people. I know, actually, a lot of you are a little bit older. Pre-cell phone, okay? You used to tell people, I will meet you at this place at that time. So if you went to the mall to meet your friends, you were going to meet at the Cinnabon at 3 p.m. When the cells are making proteins, it's the same sort of idea. You would have a small ribosomal subunit that is going to find the mRNA, and then tRNA will meet the mRNA at the start codon. So it's kind of like, here's all of the things coming together to make this process work. The large ribosomal subunit will then form over the small one, and we end up with something called an RNA complex. An RNA complex is going to be when we have all 3 types of RNA coming together. At that point, we are ready to begin synthesizing proteins. So the start codon is AUG. The transfer RNA is going to have complementary UAC, and it's going to be carrying methionine, which is going to be all in one spot on the ribosome. So how does elongation occur? Well, really, we read one triplet codon at a time, and there are tRNAs that are going to find the matching codon, and then they are going to bring in whatever amino acid it is that is with that codon. So if AUG codes for methionine, right, and GAC codes for aspartame, that's what's going to be brought in. So as we read those subcodons, those triplet codons, and we bring in those amino acids, we are linking together the carbons to the nitrogens with the peptide bond through dehydration synthesis. Okay. Hopefully all of this you're going, man, we just talked about this in Unit 1 and I know it's been a while, but like it should be jogging some of that stuff loose. This continues until we reach a stop codon. Once a stop codon is reached, termination occurs. When we release that stop codon, there's something called a release factor that causes this whole thing to come apart. The RNA complex breaks. Now, your cells are not wasteful. They are going to be doing this over and over and over again. We're not making mRNA, using it once and being done. We're going to use it until we no longer need whatever that product is. So these are going to continue to go through that process. And that polypeptide will probably go to the rough ER for folding and processing. So it's a beautiful process. Basically what ends up happening, again, in the nucleus, we have transcription, we unzip DNA, we make mRNA, and then we make it mature, okay? We then have that mRNA go out into the cell, and it's going to find a ribosome and the tRNA that matches. It's going to build out those peptides, linking together through dehydration synthesis until we get to the end. So, make sure that you can do the questions on this, check your understanding, give yourself a pause, go look and see if you can answer all of these. And then there is a diagram of the steps of protein synthesis in Canvas. I drew it for you guys. I want you to be able to take it and answer and label all the steps and molecules. So try and do that. And then when you're ready, come back to learn about what happens when the DNA is not copied correctly and when mutations occur. Okay, let's talk about some mutations. I found some fun ones because, of course, if I'm going to talk about cats, I can talk about cats, I'm going to talk about cats. So, a mutation is a change to the DNA sequence that may or may not affect a gene, okay? A mutation is just a change. Now, some mutations are really good for organisms, some are really bad for organisms, and some are just, they don't matter, okay? For instance, if you have freckles, that's a mutation. Are freckles affecting your life? No. Does it matter then? No. Is it increasing my fitness? Is it decreasing my fitness? Is it having an impact on my survival? No. So it's a neutral mutation. But if I have a mutation as a rabbit that can run faster, that's going to be a beneficial mutation. If I have a mutation that's going to lead to something not working, that's going to be maybe a bad mutation. So these can result in changes to the reading frame. And we know that mutations are important. No mutations are still going to be, it's important. We need it, okay? No mutations equals no evolution. And evolution is one of the key characteristics of life and a major theory of biology. So I did pull out an example of this American curl mutation in cats, which can result in the ears that kind of curl up and flip back. A normal protein is going to go through the normal base pair sequence. It's going to have a normal fold, and so the ears of the cats are what we would consider the normal phenotype, the normal physical expression of that trait. Now, if there is a mutation that changes the base pair sequence, it might change the amino acid that then results in the protein not folding properly. That is then going to potentially result in a mutation to the phenotype, such as what we see in these cats. And they have these little curly kind of mouse ears. I think they're adorable. Doesn't hurt the cat. It's not really a beneficial or a detrimental mutation if it's a house cat. So it would be considered something neutral, but it is something that we do see. Okay, I do like that we have also these examples, and I don't know why it's breast, but just go with it, okay? Beast, feast, breast, best, beats. Those words all have similar letters. So if beast is our normal and we substitute the first letter, we can change the meaning of the word. That's like what happens if we substitute out a amino acid. It changes the meaning of the protein sequence. If we add in a letter like the R into beast, we now have breast. That's going to also change that. That's an insertion or an addition. If we remove the A, now it's best, and that's a deletion or it's not an insertion, it's just a deletion, okay? And an inversion would be if we swap two of those amino acid sequences around. And so now instead of beast, we have beets. The basis of these words are all the same, but every word means something different because it has a different set of how those letters are formed. So when we're talking about DNA sequence mutations, what we really are talking about are point mutations or frameshift mutations. And those are going to be what is causing those changes to that amino acid sequence. So So a point mutation is usually like a single spot, and it could have zero impact. So for instance, this missense mutation is going to result in an amino acid substitution. So if I should have a T here, I have an A here now. That's going to be a difference in my amino acid sequence. Okay. Now, that one might not cause a problem. There's a lot of redundancy. If it doesn't change anything, cool. You would never even know that that mutation occurred. The same thing as like the silent mutations. It may not actually affect the functionality of that protein. But if we change out a point mutation for a stop codon, so it's called something called a nonsense mutation, I am now actually removing part of that protein, which is going to absolutely have an impact. It's like if I were to say a sentence, oh, did I not end that sentence? That's the same thing, right? I said say a sentence. There's no logical, you know where it's going to go, but if your cells don't have that protein as a direction, it's not going to go there, okay? So nonsense mutations are really bad. Silent and missense mutations may or may not cause problems. If you remember the sickle cell anemia, that is a missense mutation, okay? Causes a problem. And then we have frame shift mutations. These are generally worse because they are going to change the sequence of amino acids after wherever that change has occurred. So there can be an insertion or a deletion, and depending on where that change occurs, it's going to change the entire sequence. Okay, so I have some examples that I've made up. Let me show you here. Okay, so here is my normal sequence. I have a DNA strand of TAC, ATG, CGC, GGG, ACT. Okay, my RNA that binds to that is AUG, and if I go over to my circle here, A is in the middle, and then I follow it to the U, and then to the G, that codes for methionine, so that's my start codon. UAC, U, and then A, and then C is tyrosine. GCG, GCG is alanine. CCC is proline, and then UGA. UGA is a stop codon. So I have methionine, tyrosine, alanine, proline, stop. That would be my protein. That would be my polypeptide that was functional. Now, if I change something, so if I go from ATG to CTG, that changes my mRNA to GAC, and it actually changes my amino acid sequence from tyrosine, TYR, to aspartame. That could change the entire functionality of that protein. It might not. A lot of these amino acids are similar, so it might not change it. Like if I were saying the word meat, MEET, or the word meat, MEAT, if I were just saying it and I didn't put it in context, it doesn't really change it. That's the kind of thing here, okay? If I then do an insertion, all of these letters after that insertion are wrong. They are not what it should be. And so we end up having a shift in the reading frame, which is going to result in a completely different amino acid sequence. Where it should be alanine, proline, stop, it's now arginine, alanine, leucine, and there's no stop. This could keep going if something was continued to be copied here. So that's obviously not going to be functional. And then the same thing with a deletion, but instead of adding to that sequence, it's going to remove from that sequence. And so in this case, I have my proline still, but I'm missing my stop sequence, and I don't know what's going to happen here. It could continue going, it could stop. So when we talk about reading DNA sequences, what we really are saying is that your DNA holds that information. The mRNA is going to be complementary to the DNA, but with A's substituted out for U's, and then that is going to be what's used to code for that amino acid sequence. I think that's it. Here's your check your understanding with a little bit of a DNA sequence to complete the following, like doing some of these mutations and seeing what happens to those words. I think that's it. If you have any questions, please let me know. Have a wonderful day.